Epigenetic control of gene expression. Ever since James Watson and Francis Crick proposed the double helix model in 1953, it has been taken as fact that DNA possesses the instructions for making all parts of an organism. In recent years, however, we have come to realise that DNA is only part of the story of heredity. It is accepted that while genes determine the features of an organism, the environment can influence the expression of these genes. However, the changes they cause to the phenotype were thought not to be inherited by the offspring. We now believe that environmental factors can cause heritable changes in gene function without changing the base sequence of DNA. This process is known as epigenetics. Epigenetics is a relatively new scientific field that provides explanations as to how environmental influences such as diet, stress, and toxins can subtly alter the genetic inheritance of an organism's offspring. It is helping to explain and maybe cure illnesses ranging from autism to cancer. It is even causing scientists to look again at previously discredited theories of evolution that suggested characteristics acquired during an organism's life could be passed on to future generations. This was known as Lamarckism. The epigenome. We learned in topic 8.2 that DNA is wrapped around proteins called histones. We now know that both DNA and histones are covered in chemicals, sometimes called tags. These chemical tags form a second layer known as the epigenome. The epigenome determines the shape of the DNA histone complex. For example, it keeps genes that are inactive in a tightly packed arrangement and therefore ensures they cannot be read. It keeps these genes switched off. This is known as epigenetic silencing. By contrast, it unwraps active genes so that the DNA is exposed and can easily be transcribed, and so switches the genes on. We know that the DNA code is fixed. The epigenome, however, is flexible. This is because its chemical tags respond to environmental changes. Factors like diet and stress can cause the tags to adjust the wrapping and unwrapping of the DNA and so switch genes on and off. The epigenome of a cell is the accumulation of signals it has received during its lifetime, and it therefore acts like a cellular memory. In early development, the signals come from within the cell of the fetus, and the nutrition provided by the mother is important in shaping the epigenome at this stage. After birth and throughout life, environmental factors affect the epigenome, although signalling from within the body, for example hormones, also influence it. These factors cause the epigenome to activate or inhibit specific sets of genes. The environmental signal stimulates proteins to carry its message inside the cell, from where it is passed by a series of other proteins into the nucleus. Here the message passes to a specific protein which can be attached to a specific sequence of bases on the DNA. Once attached, the protein has two possible effects. It can change acetylation of histones, leading to the activation or inhibition of a gene. Methylation of DNA by attracting enzymes that can add or remove methyl groups. The DNA histone complex is the association of histone proteins with DNA. It is a weak association, and the DNA histone complex is less condensed or loosely packed. In this condition, the DNA is accessible by transcription factors, which can initiate production of mRNA and switch the gene on. Where this association is stronger, the DNA histone complex is more densely packed, and in this condition the DNA is not accessible by the transcription factors and is so switched off. Condensation of the DNA histone complex therefore inhibits transcription. It can be brought about by decreased acetylation of the histones or by methylation of DNA. Decreased acetylation of associated histones. Acetylation is the process whereby an acetyl group is transferred to a molecule. In this case, the group donating the acetyl group is acetyl coenzyme A, which is found in the link reaction of respiration. Deacetylation is the reverse reaction where an acetyl group is removed. Decreased acetylation increases the positive charges on histones and therefore increases their attraction to the phosphate groups of DNA. The association between DNA and histone is stronger and the DNA is not accessible to transcription factors. These transcription factors cannot initiate mRNA production from DNA. In other words, the gene is switched off. Increased methylation of DNA. Methylation is the addition of a methyl group, which is CH3. In this case, the methyl group is added to cytosine bases of DNA. Methylation normally inhibits transcription by preventing the binding of transcriptional factors to DNA and attracting proteins that condense the DNA histone complex, making the DNA inaccessible to transcription factors.